present. This is an audience that I really appreciate the opportunity to speak in front of. I want to tell you about an old problem, origins of life. This we all know about Darwinian evolution, that you came from your parents and they came from their parents, and our species came from an earlier species. And if you roll the tape all the way back to the very first cells, um, Darwinian evolution gives us a recipe for how that whole process works. What's missing is there was some kind of a singularity. This was first identified 100 years ago by Oparin and Haldane. And the singularity was somehow that biology started from simple chemistry, simple molecules, maybe on Earth, maybe from astronomical bodies. But the issue is, what was this singularity? How do we get life from simpler chemistry? It's not known, it's not been repeatable, there's no experiments. So we need theory and speculation, and there's been lots of it. These are just a few of the many books and many papers that have speculated about this problem. Did RNA molecules come first? Does DNA somehow drive itself? How do you get order from disorder? That's sometimes called the blind watchmaker. That means a watch is a highly ordered object, and the question is, how could it have gotten created from a blind watchmaker, some disorder, starting from some disorder and a process that leads from disorder? It needs a full story. Very often in this origins of life prebiotic world, of much of it is from synthetic chemists. We see little jigsaw puzzle pieces. Maybe this RNA molecule does this, or maybe this metabolic process works without an enzyme, and so on. Little pieces of a story about origins of life, but we don't have a whole story. And our aim here is to see if we can put together a full story, however, fully recognizing that many of the jigsaw puzzle pieces that we put in are going to be wrong, maybe very badly wrong. But we just would like to have a bigger tapestry, something that we can use to think about, give us a conceptual model of how the process happened. So it's not just so this is like a whodunit, I call it. That is a mystery story. We want the whole story. We want to know not only the who did it, but the how and the why. In this case, much speculation is, did RNA come first? Do proteins come first? And so forth. That's sort of the who. Those are the kind of the nouns, but we also want to know the verbs. How did it happen? What was the physical process? What are the forces, and what was it driving towards? Why did chemistry suddenly lead to something that persisted for 4 billion years? So that's what we're going to do, is to try to put together this fuller story. And the reason that we have what I think is a useful new angle here is because we think about the problem from a different point of view. Much of prebiotic chemistry is from organic chemists, and there have been some spectacular advances and insights from it. But there's another angle too, and that is it comes from the physics, thinking about the physics and the driving forces, and also thinking about proteins. What does it take to create protein sequences that could be functional, and how would we get all of this from disorder? So I show here the four main questions that we raise. One is, what is the force? Chemistry wanted to do something. Somehow, something in chemistry started heading off in a direction that ultimately became biology. It wasn't headed towards biology. What was it headed towards? And biology was perhaps the answer to it. Fitness. We have an idea in biology called fitness, and that is when two species compete against each other, the one that has the higher fitness wins. Now that's in biology. We don't have a similar thing in chemistry. We don't have molecules competing for resources. And yet, at the origins of life, we need something like a fitness function to get the whole flywheel rolling, get the ball rolling forward. How does it all come from disorder? One kind of experiment that origins of life people do is on laboratory benches. You can create certain sequences of molecules, for example. But how did the early Earth do this all by itself, by disorder processes? And fundamentally, a really important key question how sequences of biopolymers like proteins and nucleic acids lead to function, and how do you find needles in a haystack? And the reason that we felt we had something useful to offer here is because we thought about protein spaces for a long time. Our work 30 and 40 years ago was in the business of looking at the protein folding physics problem. How do proteins fold up? How do they find their native states from their conformationally disordered states and so forth? And what we have found, and what I'm going to describe to you here, is that so much of the origins of life question is wrapped up in so many of the same questions we had back then, needle in a haystack questions. How do I find some rare event in a huge sea of what seems to be just noise? Now, the starting point is to say that life has two sequence polymers. One is proteins, and the other one is, I'm calling it rDNA for short. That is RNA and DNA. Those are the nucleic acid-based polymers, and proteins are the amino acid-based polymers. And life can't exist without either one of them. So the origins of life must have had some kind of a cycle of this type, of this basic type. This is how life works today, and it must have started in that way. So, but the story that I'm going to tell you is a way that you could get started with proteins alone. We can't get this whole picture, this whole cycle, but we can get a starter that is persistent enough that it can carry forward like an evolution, like a Darwinian evolution process. That's the story that I want to tell you. So I have to start here because if I want to say something about how did chemistry turn into biology, I have to say what that boundary is. What is life? How do I define it? If I had a model, how would I know if I'm there? And so it's been difficult to define living systems. Lots and lots of definitions are out there. It's been controversial, but one that seems fairly consistent is the one that's more or less a consensus, I would say, is the one from NASA, and that is this: life is a self-sustaining chemical system capable of Darwinian evolution. There's definitions you could, for example, if you made a definition that said, well, life is something that eats and it grows and it duplicates. The thing is, a candle flame does that too. So you have to be careful how none of the simple definitions will work for what a living system is. And so I underlined this part here, capable of Darwinian evolution, because that is a part of most of the consensus definitions today, capable of Darwinian evolution, and yet it's weird. It's weird because we are trying to define a thing, namely life, not by the thing itself, but by how the thing changes. It's got to be something that is capable of Darwinian evolution. That's a little strange for a way to define something. On the other hand, it also gives us an opportunity. It says, if you have a model for how chemistry came to biology that doesn't explain Darwinian evolution, then it's not a model that is sufficient to account for what a living system is. So the first point we have to deal with is that life can't originate until it can propagate. You have to have some machinery. You have to have something like a Darwinian evolution process. You have to find a way that some kind of molecule making became sustainable. And so the question is, what is that sustainability? What is the flywheel? What is the thing that got it all started? And that's the first thing that we need to think about from the point of view of making physical models. So what was the driver? A famous book by Erwin Schrödinger in the 1940s called What is Life was very insightful and raised the question of how would chemistry ever beat the second law of thermodynamics? And the whole book was really puzzling over that. And at the end, he didn't answer it. You know, this is 1944. Would be not expected. That's way before even the DNA molecule structure was known and much about biology was known. But it was super insightful for the day. But he raised the question of how would you ever beat entropy? And his focus was really on equilibrium. He called it negentropy. That negentropy decreases. And And the question is, how do you beat that? And so he raised the question of, do you need new physical laws? And the argument I want to raise here is, no, you don't need any new physical laws. It's just that you have to recognize that whatever created life and how life runs now is by non-equilibrium. And what you have to recognize is those are driven systems, and driven systems are not ruled by the laws of equilibrium. They're not tendencies towards equilibrium, they're tendencies towards something else. And I show that in the bottom picture here with a wall plug. And that wall plug is just a little metaphorical toy indicating that you got to have some input from something that's driving. So for example, think about a television set or a computer. You plug it in and it does things that are truly remarkable from the point of view of if you thought only about the second law of thermodynamics. It's not going to equilibrium. They're plugged in. A television set is a system that is wired in such a way that it performs very interesting and complicated steps, and it does that by virtue of being driven. And so being driven means there's lots of physics that is pretty unexplored, and we need to explore it here. We need to find how the drivers and early Earth, the pumping process of some kind from the environment, how did that couple to being able to make molecules in a long-term persistent way? So let's start here with what is evolution today? What is biological evolution? And how could we use that thinking about what it is to roll the tape backwards and see if we could figure out what it might have been back in the early stages? So on the right-hand side, I show this little picture. We call evolution a machine, a maker machine. And it's a machine because it goes through a series of steps, A, B, C in time, and then the next time unit and so forth. Goes through the series of steps, and it does it because it's driven. And the series of steps happens to be a search, compete, and select process. That's what Darwinian evolution is. This is today. And today, here's how it works, is at time t, you have a population of cells, I'm calling them X. And then at the next interval, what happens is a mutation happens in one of those cells. We call it a momsomble because it is an ensemble of possible mutants in a molecule, and because the mutations are in maker molecules, and the makers here are moms make more moms. That's in biology. And so you have a wild type population, you have a mutation. The mutation then grows up and becomes a population Y. Now the population X and Y compete with each other, and one of them wins. And whoever is the winner gets now more resources for the next round at time t+1 and goes forward from there in the same way as before. We call this a maker machine. It's Darwinian evolution. And again, it's a machine that it's not a device that does a deterministic thing. It's a device that does a search and compete and select thing. So on the left here, I describe that in a little more detail. It's about making. Makers make more makers. But it's about mutations. It's about change. It's about heritable variation. That's what Darwinian evolution is. It's about making changes. You are not exactly your parents. You're something like your parents, but you're not exactly like them. And it's the not being exactly like them that's very, very important here for evolution to be able to adapt to different environments. Then you compete among the makers. The alpha dogs win, and the alpha dogs get more resources, so they go forward. This is the alpha dog. In this case, is the population Y, makes more alpha dogs. This is Darwinian evolution. What we're trying to figure out is, could this process have started in some way prebiotically through simple molecules? I'm only going to show this briefly, this one math slide. It's just to indicate how we make models. First of all, the basics of this model go way back 100 years to the Volterra-Lotka population dynamics models. There are lots and lots of these things, and they're used very commonly in evolution and ecology textbooks. But we need some alterations to these basic models in order to think about molecules. For one thing, cells are self-contained, organisms are self-contained, so they have an identity. Molecules don't. molecules are just floating around and they have concentrations, but they won't have boundaries at the beginning. So we have molecules before we have cells, and that means we need to treat the density differently. Second thing is you can't just have pure Darwinian evolution, winner take all, because winner take all is too brittle. Darwinian evolution would have died out a long time ago if it's only winner take all. Because think about it this way, you have the orange team and you have the green team. Now the green team wins under one environment. Now the environment changes, the green team gets killed, and nothing can go forward. You have to have some coexistence in a model. So this model accounts for that as well. And the last point is birth and death is the basic process that underlies any of these population processes, but it can't be linear. Often in population dynamics, you take the linear assumption. It can't be linear in early stages, and I'm going to show you why in just a second. So now I've simplified this, and on the left I have a picture. DADTA is some population of moms, of makers, let's call it, molecules in our case, as a function of time. Now, a normal birth-death process is the first term here. There's a growth term, a growth coefficient G1, and there's a death term, D. And death usually wins in chemistry. That's the Schrodinger problem, thinking about the second law of thermodynamics. D is always bigger than G1, and so chemistry always goes to equilibrium and it dies out and it doesn't continue to propagate. Now, the point of showing you that population dynamics, however, is that when you do now what's called an invasion analysis or an extinction analysis, what you can show is that the lowest order Taylor expansion beyond simple and birth and death has a next higher order term. G2 is a coefficient and A squared is the mom population squared. If you don't have this cooperativity term, we call it cooperativity. If you don't have this cooperativity term, you can never escape chemistry. And this is what Schrodinger was missing. If you just think birth and death processes, you never get out of chemistry. And here's the picture that shows you that on the right side. Let's start with G2 equals 0 down here at the bottom. G2 equals 0, that's normal birth and death. What happens is, well, if G1 is if the growth is too small compared to death, I just live in chemistry always. Everything goes to equilibrium. If G1 is greater than the death rate, I live in biology. That is, moms are making kids, or making kids, or making kids, and the growth wins, and the death loses out, as long as you have a number of kids is proportional to the number of moms. Now, the problem is if you have G2 equals 0, you have no cooperativity, you can't get across this wall. You can't get from chemistry to biology. With this cooperativity term, it turns out you can go into a region, we call these fold cats. I'm going to explain that in a minute. I'm going to show you a mechanism. But you can get into this region where this wall has a very small barrier and you can fluctuate across it. So if I have a cooperativity term, I can get up here, I can cross over into biology, and now I'm irreversibly in biology. This is the process we care about. Here's what it's saying. Classically, we think about growth and death as being simply one or the other is dominant. And that means moms are making more moms. What we need here to get from chemistry to biology is moms have to make better moms, have to make even better moms, have to make even better moms. That's what I mean by cooperativity. That has to be true for a while. And once you keep making better and better and better moms, now you can get to a point where biology sustains, and then moms can make moms can make moms. You got to have this cooperativity term. And now I'm going to spend most of the rest of the time talking about where might that come from in a microscopic mechanism. So what are the molecules that might be involved and what are the mechanisms? I'm going to focus on protein molecules because that's really evolution's fuel. Darwinian evolution functions on by selecting on phenotype, not genotype. And what those terms mean is phenotype means actions and mechanisms and how and processes and how they work and speeds and rates. And I grow faster than you do, and that's what phenotypes are all about. Genotypes are all about the information stored in RNA and DNA and so forth. But biology for Darwinian evolution, what we select on is it you or me who grows faster in a given environment? And the business of function and the business of action is what today protein molecules do. So evolution's fuel, the way it makes choices, you can do make random mutations, but then to compete and do selection and make choices. Those choices are based on proteins, not on the RNA and DNA to first approximation. Protein and how is it that proteins are so special compared to RNA and DNA? It's because proteins are able to form actions because they're able to fold. This is the pipeline of information, if you will. You have an amino acid sequence. Each of these beads is representing one amino acid in a protein molecule chain. And I'm giving them as two colors here. The red beads are hydrophobic, meaning they're kind of oil-like. They don't like water. And the blue ones are polar, means they do like water. And the amino acids in today's 20-letter alphabet of amino acids falls into these two classes about evenly, 50-50. Now, the argument I'm going to make here, and is now known pretty well from proteinology, is that it's the sequence of amino acids that dictates that it folds into a compact structure of a particular shape. And in particular, what it does is it's largely a binary code where the hydrophobic ones, the oil-like amino acids, don't like to be near water. There's water all around here, so they get buried away in a core. And then the polar ones are mostly on the outsides. So a given sequence folds into a given fold, and that given fold has a molecular function, like a molecular motor, say, or like a protease. A different sequence folds into a different fold, has a different function. And my argument is proteins are the central actor in evolution because they are able to fold, and that means they are able to function. And the informational pipeline is clean. I have a given sequence, gives me a particular structure, which gives me a particular function. Now I'm going to tell you about needle in a haystack problems. I'm going to tell you about two of them. One is the old protein folding problem. And I say old because it's now solved. There's two protein folding problems. One is really familiar from most of the literature. There's an algorithm called AlphaFold, and what it does is it takes amino acid sequences and it will tell you the native structure of the protein. That was a very important computational problem. 3 Nobel Prizes last year in chemistry for figuring out AlphaFold, for figuring out how to take an amino acid sequence and figure out the native structure of the molecule. The physics protein folding problem is different, and this one was solved by a smaller community of folks in statistical mechanics. And this one is all about, here I have a haystack of conformations of a single protein, and in the denatured state, like at high temperatures, for example, or when they first are made biologically, the protein molecule has a large set of conformations, and it's ultimately it folds to find its native structure, and it can do it in a very short period of time. But this haystack of conformations is huge. If I have 3 different rotational isomers around each bond, and I have 130 amino acids, a typical protein, then I have 10 to the 62 as the size of my haystack. I'm looking for one structure. People represent that as a golf course landscape. You're searching a very flat surface that's huge, multidimensional, thousands of dimensions, and it's going to look for a single hole. That was the needle in a haystack problem of protein folding. Here it is for sequence space. Each amino acid could have any one of 20 different amino acids. My haystack now is sequence space, not conformational space. I have 20 possible amino acids at each position. Now I have a huge haystack, and it looks similarly. How do I find the needle in the haystack? We now know the answer to this. One is it's the binary code, not a 20-letter code. So that means already I lose, if n is 130, it means I lose 130 orders of magnitude of size of the haystack that I need to search. This idea that there's a binary code, which came from some lattice models we and other people did back in the late 80s and the 90s, was validated by experiments. It turns out you can swap out amino acid sequences in real proteins, and the binary code gives you the right answer. And most importantly for here, the search to find the native structure is not a flat landscape. It's not a golf course. It's a funnel. And so you roll a ball in a funnel and it will go right to the bottom. And so it's because there's cooperativity, you take one step down and it helps you get to the next step down and to the next step down and so forth. Making long chains is very hard. Prebiotically, lots and lots of experimentalists have tried to do this. Could I make amino acid sequences? And you end up with this so-called Flory problem. This is a log scale. This is chain length. Almost all of the chains are very short. Very few of them are long. This is the thing we need to solve. But it turns out that these largely polymer science focuses on either all red bead chains or all blue bead chains. But when you think about proteins, these are sequences of red and blue beads. Biology has the ability to string them together in specific sequences. Polymer science usually has, in the past, has mostly just been able to make random sequences or all blue and all red. But if you have the ability to make sequences, then some will fold like proteins do, and some will catalyze. They'll have surfaces on which reactions can happen. We call these yarn balls, HP chains. If I just make random chains, I have a macroscale catalyst here. Suppose it's some rock in a lake in Nebraska somewhere, and I've got hydrophobic and polar amino acids. They will make stuff like this. They'll be just sort of little random gamishes, but because the red beads are hydrophobic, they're going to collapse down a little bit. Now, what we know from computer modeling is that's enough to solve the problem, is to have these HP sequences and have specific ones. Here was the plot I showed you before, the Flory problem. I can't get long chains, but with these HP sequences, I can get long chains. These are populations out here that are 20 mers and 25 mers. They have specific sequences they can fold, and the autocatalysis, the G2 term we're looking for, why is this a flywheel towards anything better? It's because, for one thing, when these collapse, they protect their hydrophobic cores from degrading by water. And that means it slows down the death process of degradation of the proteins. And secondly, they catalyze. Here you see a little catalytic surface, and they can elongate other chains. Certain chains can elongate other chains. In this model, in the computational model, what we see is this is a case where you have essentially, I make these random chains on this rock in Nebraska. I mostly make just nothing but infinite amounts of junk, small chains that are random. They don't do anything useful or interesting, but a few of those fold up a little bit, and a few of those fold up more and fold up more. And the thing is, as long as it's being powered from outside, as long as you continue having a supply of amino acids and that catalytic rock somewhere, it will create a few longer and more functional molecules. Let me skip this. We can test this by experiments. Do random HP chains collapse? That's part of what the mechanism would predict. Can they catalyze the formation of peptide bonds, and do they gain length and stability and function? I don't have an answer to the third one. I do have some results on the first two. We do this work. This work is done in collaboration with Ron Zuckerman, who invented these polymers called peptoids. That's not a typo. These are different than peptides. And Donghui Zhang. These are molecules that are very much like peptides, like proteins, except that the side chain group on a peptide is pulled off of this carbon, C alpha carbon, and plugged onto the amide nitrogen. Now, it has a lot of very different properties, but in many ways, it's very similar to protein. So these are protein-like molecules, except they don't fold to protein-like states. They allow us to study the protein-like features of what early proteins might have been like, the disordered ones, the ones that have slightly interesting sequences, but not really biologically useful sequences yet. We can study properties of ensembles of the early proteins by using these peptoid molecules. We have total control over everything: chain length, composition, hydrophobe type, and so forth. And briefly, the remarkable result from all of this is that whole space, the whole yarn ball space, all of random sequences, red beads and blue beads of a given chain length, let's say 50 mers, act like they're folding protein. This is a picture of an actual single protein and in a small angle X-ray scattering Kratky plot. And what it shows you is you have in the unfolded state, you see a sort of a plateau-like region. In a folded state, what you see is a dip. And I'm showing you here a whole random space, the whole peptoid space, the yarn ball space, all hydrophobic and polar sequences of 50-mer chains, 50-50 HP composition, and it shows exactly what you expect of a folding protein, that when it's all polar, it has no ability to make a core, then you get nothing but floppy chains in solution. If you make it 50-50 hydrophobic and polar, then you get something that looks exactly like a folding protein, except this is not one molecule, this is a whole space. So what it's saying is prebiotic disordered order, we're starting from a disordered state of the entire sequence space, and yet it's already looking like a protein. It also denatures. If you put it in a small amount of denaturant, you see this, its native-like structure, compact-like structure. You put it in high denaturant, what you see is this unfolded structure, just like a real protein would do. I'm not going to go through this bottom part. So this means these yarn balls are compact, and they do fold up to some extent, probably very loose, not like native proteins today. The question is, could they also catalyze reaction? One way they could do it is by localizing reactants on a surface, let's say. This is a picture of a yarn ball, each of these things. They could expose side chain gardens. Proteins have amino acid side chains, and they could provide small nonpolar spaces for reaction. So how might you assist in peptide bond formation, for example? This is an amino acid. This is another amino acid. They're individual to begin with, and when I make a chain, when I grow a chain, a peptide chain longer, I have to remove a water molecule. So anything that helps remove a water molecule can help promote this reaction. In water, this is an uphill battle to try to get these two things to come together to form a peptide bond. But in methanol, they will come together and form a peptide bond. And here what we're doing in this experiment, what we do is we take these two monomer units it's a dehydrolysis reaction, just like peptides. Take these two molecules, they need to exclude water to come together to form a bond. And when we put our yarn balls in there to act as little reaction vessels, it turns out it does accelerate the reaction. And these are random. This is the whole yarn ball space. So it says this space is already starting to act like proteins in a highly disordered world. Let me briefly go through this. Many of you may not care about this. This may be a little subtle. Could we have argued that the RNAs could have started? The predominant view out there at the moment is there was an RNA world, meaning RNA molecules might have been sufficient on their own without proteins to start origins of life. Let me just skim over this. I can come back to it later if anybody's interested, but the bottom line is RNAs are not very good at being functional. There are some that are very good at function, but for the most part, as a matter of material science, RNAs in general don't have structure and function. And I'm going to skip that too and just say, what have we got here so far? So the argument is that fold cats set the table for origins, meaning that they can start the flywheel of a Darwinian evolution-like process. They can lead to better molecules. They can lead to longer amino acids. They can lead to proteins that become catalytic. But it doesn't, this is only the beginning of a story. This is not an origins of life story because it doesn't say, how did I get a genetic code? And how did I get RNA and protein to come together? And how do I get metabolic networks? And how do I get cells and membranes? But what it does do, that's the important step that you need here, is it tells us how to get a first form of fitness. And that first form of fitness can now help get subsequent forms of fitness. What is the first fitness? The first fitness is just persistence. How long does a molecule live in its environment? And different amino acid sequences live different amounts of time, and that's partly because how compact they are, how well folded they are, and how catalytic they are. So what this gives us is a fitness function that works in chemistry that can help get us out of chemistry by those forms of cooperativity and into biology. The one thing I will say is that even though this foldcat story really is only kind of the setting the table here, it does satisfy NASA's definition of life, which is that life is a self-sustaining chemical system capable of Darwinian evolution. Now, my argument about that is not that foldcats is the true origins of life, but rather that the NASA definition needs to be upgraded. So, summary, our whodunit story is about foldcats, and the who is protein molecules. This is what gets it started. It started by making random short peptides. This is just a garbage generator. And it's just that because you have a process of generating different sequences and then competing them and selecting them, it's that there are some good ones that make it through. As long as you keep pumping the system, you keep getting some that get better and better. A very small fraction, but what starts it is just making these little random short chains. It's a disorder to order process. The positive feedback, the autocatalysis, the G2 term, the cooperativity that you need, where does it come from? It comes from the fact that these random HP sequences, because they have hydrophobics in them, will fold a lot. They'll be compact in water. This is true for half of the sequence space. Half of the sequence space has lots of hydrophobic residues. They'll be compact. And if they're sufficiently compact, they'll have stable surfaces that can catalyze reactions. The action is the Darwinian evolution machine action, which is sample, compete, and select. The physics is really simple. It's just oil-water physics. It's oil-water physics, but in a polymer. That is, you have hydrophobic things that don't like water. You have polar things that do like water. But if you string them together into particular sequences, it turns out that will cause a chain to fold into very specific structures, native structures. And that's where you get off and running now into having protected cores, catalysis, and being able to keep walking up fitness landscapes. The goal of all this, why did chemistry rise up and go to biology? The goal of all this was not because it was seeking biology. It was just trying to seek stability in an environment, just persist in an environment. Certain things can persist longer than others, and they have to be able to persist in fluctuating environments. You don't know what your environment's going to be tomorrow. And these molecules have to be able to out-survive each other in whatever environment they see. And it was just these persistences that ultimately the system kept getting more and more persistent. And it just turns out that the biological molecules that make that happen today are ones that are great molecules for ensuring persistence. So a big picture final slide is that evolution discovered proteins, which really rewired the world of what chemistry and physical chemistry was happening 4 billion years ago. It rewired the world because it is a single process, the making of a protein molecule. You have some kind of a catalyst and you have monomer units, and that single process creates a vast space of catalysts. They're just all different sequences, but the process is the same. There was an untethering. Once these catalysts became good enough, these yarn balls became good enough, there's an untethering that must have happened. This would have been a really transformative event when you disconnect from that rock in Nebraska, and now catalysts are editable and they're miniature and they are freely able to spread out through a much broader medium. And also there's the working together. Proteins interacting with other proteins are fantastic at doing all the jobs of biology. And that's it. These are the folks that did the work, and thank you all for listening.